Hello National 5 students, welcome to this very short video on the stages of genetic engineering, which you know is a tricky topic from Unit 1 of the National 5 biology course. I've had a little request to do this video, so we're going to run through the steps involved in genetic engineering so that you can learn that for your exam, and when that question comes up in the exam you can feel much more confident. So genetic engineering is a process where we're transferring genes from one organism to another. Okay, and we use enzymes in this process. That's an important learning point to remember. Enzymes are used throughout this process, and we'll talk about where they are in our example here. Okay, so we've got a nice diagram that shows us the stages here, okay, visually. So we've got our required gene and our plasmid. Sometimes in the exam they ask you to label these things or identify these things, okay? So you need to know what the plasmid looks like within the bacterial cell here, and where the required gene is found, which is on these structures called chromosomes. Okay, so we need to find the required gene and the plasmid, make sure we can identify both of those things. So the stages, we'll go through them just now. The first stage, we need to identify the section of DNA, that's up here, that contains the required gene from the source chromosome. Okay, so this here is stage 1. We identify the required gene from this source chromosome. Stage 2, we need to extract that required gene. Okay, so we remove that or extract it using enzymes. Step 3, we need to extract the plasmid from the bacterial cell. Okay, so extract that plasmid from the bacterial cell. That happens up here. Okay, we've extracted that plasmid from the bacterial cell there. Step 4, we need to insert the required gene into the bacterial cell. Okay, so that happens right here. Okay, now again, we need enzymes for this part of the process to cut open the plasmid and to insert the gene. Okay, so wherever we see extract or insert, we need enzymes for that part of the process. Okay, lastly, we insert the plasmid back into the host bacterial cell to produce a genetically modified organism. Okay, that is this stage here, where we've put the modified plasmid back into the host bacterial cell. Okay, that's the host bacterial cell. Students often make the mistake of saying put the modified plasmid back into the bacterial cell, but you must say into the host bacterial cell. Okay, so this is the process and the steps involved in genetic engineering. Hope that short lesson has helped you and that you've learned a little bit. Leave some comments below to say if it has helped you, or if you have any further questions, or if you have any other topics that you'd like me to do a short video on. Thanks for now, folks. Class dismissed.